a groundbreaking antivenom, capable of neutralizing the deadly toxins of 17 African snake species, has emerged from scientific research, offering a glimmer of hope in the fight against snakebite fatalities. With over 300,000 snakebite incidents annually in Africa, resulting in thousands of deaths and countless amputations, this new development is a beacon of progress. The traditional approach to antivenom production, involving the exposure of horses to specific venoms, has its limitations. These methods often lead to allergic reactions in patients, and are only effective against a narrow range of related snake species. However, the innovative nanobody-based antivenom developed by researchers presents a remarkable advancement. This is a significant leap forward in synthetic antivenom development, says Juan Calvite, director of the Evolutionary and Translational Venomics Laboratory. He acknowledges the potential impact, but also highlights the challenges, particularly the cost of production, which could hinder its accessibility in poorer regions. To create this revolutionary antivenom, researchers employed a unique strategy. They exposed an alpaca and a llama to venoms from 18 deadly African snakes, including cobras, mambas, and the rinkhouse. In response, these animals produced special, tiny antibodies known as nanobodies. The compact nature of these nanobodies allows them to swiftly diffuse through tissues and bind to toxins in hard-to-reach areas of the body. The researchers collected the blood of these animals and identified the nanobodies that bound most effectively to the venom toxins. These nanobodies were then produced in the lab and tested for their ability to neutralize the venom's effects. The result? A powerful mix of 8 engineered nanobodies, forming a highly effective antivenom. In laboratory tests on mice, this nanobody serum successfully prevented death from 17 of the 18 targeted snake venoms. The only exception was the venom of the eastern green mamba, which was not fully neutralized. Further analysis revealed that the antivenom effectively neutralized 7 toxin families present in the venoms, and reduced tissue damage caused by cell-killing venoms. The new antivenom outperformed traditional horse antibody-based serums, with mice treated with the nanobody mix surviving multiple venoms with fewer symptoms. Andreas Hugard-Lossenkiel, a biotechnologist and senior study author, emphasized the significance of their work. Our main achievement is demonstrating that an effective recombinant antivenom can be created with a surprisingly small number of nanobodies, outperforming existing ones. The potential for large-scale production of this antivenom is exciting. Lossenkiel explained that it could theoretically be manufactured in bioreactors, independent of snakes and horses. However, further testing is required to determine the optimal dose for human use and to scale up production. The team is also exploring the use of these nanobodies against Asian cobra venoms to develop cocktails with broader geographical relevance. The concept of a broad-spectrum or universal antivenom is gaining momentum. A notable study in 2025, published in Cell, utilized human antibodies from a snakebite survivor to protect mice from multiple cobra and mamba venoms. Yet, practical and economic barriers remain, making the development and affordable manufacturing of such an antivenom a challenging endeavor. Calvite praised the new nanobody venom as a significant advancement but cautioned that the dosing requirements for human treatment could be complex. He suggested that a therapeutic dose to treat envenomings from all target snakes might require up to 50 grams of nanobodies, a quantity that would significantly impact production costs. In conclusion, while the new nanobody mixture holds promise as a safer and more scalable snakebite therapy, further testing, manufacturing optimization, and regulatory approval are essential before it can be administered to human patients. The journey towards a universal antivenom is a complex one, but with continued research and innovation, the future looks brighter for snakebite victims worldwide.